Thank you. So I'm a master's student at the University of Oslo, and I will present my project idea today. So I don't have any results yet. So in my project, I will basically compare synaptic protein concentrations in stress-resilient and stress-susceptible red brains in a model of depression, which is called learned helplessness. So here's just a quick overview what I'm going to talk about. So major depressive disorder, mind wandering, the default network, and synaptic plasticity, and how this is all linked to my project. So major depressive disorder affects between 4 and 15% of the human population during their lifetime. And of course it affects the persons individually, but it also generates really high costs for the society. And one thing which is very common in depressive patients is that they do a lot of negative rumination. So they're kind of stuck in their negative thoughts. So what then could be different when you compare it to controls might be what they do in their resting state. And what is the resting state? So when you're just like sitting around and just thinking about whatever, which is not dependent of the environment you're in right now. So one thing you do is, for example, that you have, when you concentrate on this presentation and you have some thoughts which are totally not linked to what I'm talking right now, you do like, those are self-generated thoughts. And when they compared MDD patients with controls, so when they should concentrate on an active task, they had not only more and more negative of those thoughts, but also they were less varied depending on the severity of the symptoms. And the network which is activated during resting state is the default network. And also there they have found a difference in MDD patients, that it's like hyper activated. And then they also found structural changes in MDD patients, for example in the prefrontal cortex, that there's a lower density of glutamate receptor subunits. So just at a glutamatergic synapse. And so I will look at rats. And how do I get rats depressed? They were put into a box where they got random inescapable foot shocks. And after that, they were put in another box where they could escape those shocks if they wanted to by pressing a lever. And if they didn't do that within 20 seconds, then this was counted as a failed trial. And if they failed more than 10 times out of 15, those were called helpless, and those showed like depressive symptoms. And if they failed less than 5 times, those were more resilient. And those two groups were bred for at least 30 generations, so that you had kind of 2 different strains. And those are the regions I will be looking at. So the primary motor cortex and primary somatosensory cortex, it's as a control, so I don't expect any changes to happen there. While the hippocampus and the cingulate cortex are part of the default network, and there I hope to find a difference between the stress-resilient and the stress-susceptible red brains. And those are my proteins of interest, so it's basically just glutamatergic synaptic proteins. And I will analyze it with Western blot. I'm not sure if you're familiar with that, but it's just like looking at the relative concentration difference and then comparing those with each other. And just to sum it all up, so MDD has, or depressive disorders in general, have a really high prevalence in our society, and the mechanisms are still poorly understood. But the default network might play a central role in this disorder, and just finding out what kind of changes occur there might help to find new medication, or just to see why are they more resilient or why are they less. And I want to thank my supervisor Sven and the rest of my research group, and Barbara Vollmeyer for giving us the rats, and Umar for helping me with some methods. Yeah, thanks.